Oops. All right, can you guys see it now? Okay, cool.
You're good, yeah.
All right, well, hi, my name is Nina, and today I'll be presenting the associations between endometriosis and the gut microbiota. So just so we're all on the same page here, we'll go over some of the terminology associated with this study. So for those of you who don't know, endometriosis is a condition where the endometrial tissue, which is this uterine lining, grows outside of the uterus. It can grow in places such as like the ovaries, fallopian tubes, or even the intestines. So it's a pretty painful kind of condition, and it affects an estimated 10% of women of reproductive age. So symptoms include, severe cramping and pain, but it can even advance to infertility even. In fact, the American Society of Reproductive Medicine estimates that of women who are infertile between 30 and 50% actually do have endometriosis. So it's a pretty big deal. And it's really not studied as much. So the gut microbiota is the collection of microorganisms, which is mainly bacteria, that reside in the digestive tract. And finally, 16S ribosomal RNA sequencing is this genetic technique sequencing the 16S RNA gene that codes for the small subunit of the ribosome found in prokaryotic organisms, mainly bacteria, for this study. So this study had some pretty advanced statistics, actually, that I even had to look up to learn more about. So we'll start off with the Mann-Whitney test. The technical stats definition is, you can see here, a non-parametric test of the null hypothesis that with randomly selected values X and Y from two populations, the probability of X being greater than Y is the same as the probability of Y being greater than X. But in layman's terms, this is just saying it's testing to see if two samples are likely to come from the same population. The Spearman coefficient test is a non-parametric measure of the rank correlation, and this is essentially just measuring the strength of correlation between two variables. The Fischer's exact test is another form of a statistical significance test, which is just used to determine if there are any non-random associations between any two categorical variables. The Bray-Curtis dissimilarity index is just a number used to quantify the differences in species populations between two different sites. For this study, it was used to detect the differences in the microbiota composition. The Shannon diversity index is another number that's just used to quantify the diversity of the species in a community. And this study was used to analyze the diversity of the sample genus of the bacteria. And finally, the ANOVA test, which is more common, just the analysis of variance, hypothesis testing, and it's just another statistical significance test that measures the variance of multiple samples, and it's used to determine if a result is statistically significant at all. So the study is important because in the last couple of years, it has been thought that the gut microbiota plays a role in a lot of different diseases, and almost any disease, actually. There's been a lot of research about the gut microbiota in general, but there has not been many studies about endometriosis and the gut microbiota in particular. And endometriosis, again, has links to infertility, and these links are unclear. So any research we can get about this disease is good. The methods are pretty straightforward from this study. They took 66 women who had endometriosis, and each of these endometriosis patients, they were matched with 3 controls. This is 198 women total for the control group. They were given questions about, socioeconomic status, medical history, including GI symptoms, things like that. They collected stool samples, and these were stored until the microbial DNA could be extracted, which was extracted using the 16S ribosomal RNA sequencing, as discussed earlier. To go a little more into the actual sample and the composition of the sample, So just the characteristics of the entire sample, the basal characteristics. The median age of the participants was 37.8. The median BMI was 25. So 95.4% had education from any secondary school, university, with 83.4% currently in school or they're working. 84.8% were non-smokers, and 63.6% consume less than 1 glass of alcohol a week. 40.9% had greater than 1 hour of exercise a week. And then in general, there are more patients with endometriosis than the control group who are being treated with hormone therapy or analgesics. This is an important note, I guess, because endometriosis is actually associated with estrogen and estrogen levels, and high levels of estrogen have been, kind of linked to the pathogenesis of endometriosis. So it's kind of a double-edged sword here because hormone therapy is really important for controlling other diseases pertaining to women's health, like ovarian cancers and things like that. But there's this new association with endometriosis that might cause other problems. So moving on to the characteristics of endometriosis patients. So 40.9% had isolated ovarian endometriosis, meaning it didn't really spread to other places outside of the ovaries. 27.3% had some involvement with the gastrointestinal tract, and 48.8% were currently being treated with estrogen or combined oral contraceptives, kind of discussed before. 46.3% being treated with progestin, same thing. 19.5% are being treated with GnRH analogs, which is just gonadotropin releasing hormone agonist, and it's another medication that affects the gonadotropins and other sex hormones. And 86.4% had reported GI symptoms in the previous 2 weeks before the study took place. So the biggest thing to take away from this is that you can kind of see that, besides endometriosis, these people are like pretty healthy, decent BMI, they're young, they're coming from decent socioeconomic status, they don't smoke, they don't really drink, they exercise. So it helps them make more accurate conclusions when there are not other medical factors that could be confounding. So they kind of narrowed it down to a more healthy population. So for the results, the ANOVA test concluded that there was significantly higher species diversity, bacteria species diversity, in the control group compared to the endometriosis group. They were able to, identify 19 different gut bacteria at the genus level. They differed in abundance between the control and the patient group. These come from the classes Bacteriota, Clostridia, Choriobacteria, Bacilli, and Gamma Proteobacter. So there were 2 bacteria from the Bacteriota class and 2 from the Clostridia class that were observed in higher abundance in the endometriosis group. And there are 2 other bacteria from the Bacteriota group and the Clostridia group classes that were observed in lower abundance from the endometriosis patients compared to the control group. There were 2 other bacteria in the Bacteriota group and the Clostridia classes that were again found in lower abundance in the endometriosis group compared to the controls. So there are another genus belonging to the Bacilli group, another genus belonging to the Choriobacteria class. They were found in lower abundance again in the endometriosis group. And again, there's another genus in the Gamma Proteobacter group that was in higher abundance compared to the control group. Another important finding is that the isolated ovarian endometriosis had a higher abundance of bacteria from the Clostridia and Choriobacteria classes compared to endometriosis that was more delocalized. And any gastrointestinal involvement had a higher abundance of bacteria from the Lactococcus group compared to endometriosis without any GI involvement. And finally, patients who are on hormonal treatment had a higher abundance in the Clostridia and Bacteriota classes compared to patients that were not being treated with any form of hormone therapy. So this plot in figure 1 is depicting the Bray-Curtis dissimilarity index with the control group versus the endometriosis group. So we can recall that the Bray-Curtis dissimilarity index was used to detect differences in the microbiota composition. This plot's actually depicting some fairly advanced statistics. What it is showing, I believe, is called a non-metric multidimensional scaling. And this is a process used to represent the original position of species communities in a multidimensional space, using a reduced number of dimensions that can be easily visualized. So it's similar to principal component analysis, if you guys are familiar with that, which is reducing the dimensionality of large data sets. Uses some pretty advanced linear algebra to do this. So you have a really big data set and you're able to create smaller variables with the information from the original data set. And then you can calculate the percent of variance explained by these new variables from this process. So what this graph is showing right here is the two principal components on the axes, which account for the highest variance, and it's plotted with the original data set from the controls and the endometriosis. And what you can see here, the ellipses, I believe, are also confidence interval. So what you can see here is that although the endometriosis group has a higher confidence interval, you can see that the control group has a lot more data spread out among the variance explained. So this figure is a little easier to digest. It is just a simple box plot with the Shannon diversity index for, again, both experimental groups. The Shannon diversity index, just a reminder, was used to analyze the diversity of the sample genus samples. And again, you can see the control group had an overall higher both mean and median of the diversity index, which implies that the sample genus was more diverse, kind of confirming what the other figure showed. Finally, this is another kind of straightforward thing. It's just a stacked bar chart of the 19 statistically significant differing genus samples with their mean relative abundance for both the experimental groups. I added these arrows right here. It looks pretty similar on first glance, but there are some differences in relative abundance that I was able to kind of show with these arrows right here. And these small differences really do add up to change the entire microbiota composition. So important things to note that the diversity of the gut microbiota was significantly higher among the control group compared to patients with endometriosis. And generally speaking, it is more desirable to have a higher diversity of bacterial species in the gut, just it helps with the proper functioning and whatnot. So again, the differences in the abundance were determined to be from the bacterial classes, the bacilli, bacteriota, clostridia, choriobacteria, and gamma proteobacter. Another thing is important to do in research is to compare your findings to other current literature, called a systematic review. So this study did it from 2020. And at this point, there were only 13 clinical studies that were studying the connection between endometriosis and the gut microbiome. And the microbiome in general, Only 6 of these studied the gut microbiota, and only 1 of these actually used the human gut microbiota. So it was really hard for them to put their literature in light of others. So the one study that did use the human gut microbiota, they found that women with late-stage endometriosis had an Escherichia shigella dominant gut microbiome at the genus level, but none of the endometriosis or the patients without endometriosis did not have the same dominant gut microbiome composition. But again, this is only one study, so it makes it hard to make conclusions and accurately compare findings. Again, another reason why is because the study that I'm presenting now really did not identify any Escherichia or Shigella dominant bacteria using their sequencing methods they used. But on the other hand, there were a couple animal studies using rhesus monkeys and mice that do suggest that endometriosis may impact the gut microbiota. However, these are animal studies, and again, the gut microbiota composition in general is still not the same as humans. So another important finding is that the results did not suggest that the location of endometriosis lesions is related to the gut microbiota. So really where the endometriosis spreads is not necessarily controlled by the diversity of the gut microbiota. But it is still hard to determine from the study whether if the gut microbiota impacts endometriosis or if it's the other way around, whether endometriosis is what is affecting the gut microbiota. So again, there's a lot of unknown and a lot more research needs to be done. So limitations of the study. Theoretically, some of the control groups, they could have endometriosis, but they just aren't experiencing any symptoms, but this is probably pretty rare, not likely. But they still are at risk of developing endometriosis because they were fairly young women. the researchers only use 16S ribosomal RNA sequencing, which they admitted in their study that this is really not a good comparison for the entire gut microbiota genome data. So in the future, more sequencing techniques should be utilized. They also could not control for the diet of their sample population, and the diet is also an important factor that controls the gut microbiota composition. It was really hard for them, to get patients or, participants with the exact same food habits. And finally, again, there's lack of similar literature, so they could not place their current work in the body of knowledge. So yeah, a lot of research needs to be done still. But are there any questions?
So do you think, so endometriosis obviously has high links with estrogen, right? And some birth controls have effects on estrogen levels. Do you think, I mean, I'm not too sure if this study covered it, but do you think it'd be worthwhile to say, hey, if we have a sample population that's on an estrogen-based birth control, how does it compare to the control, and how does the gut microbiota work with that? What well, what do we see in there, and do you think there's different levels of it? Do you think that would be like a next step further, or do you think that's not really the path we should go?
Yeah, I think it's a really good next step. Part of what this principal component analysis attempts to do is, so when I mean dimensionality, I'm talking about the other factors like their health status, including the hormone therapy. So like that, it's hard to track which principal component is really explaining that variance. But for the future, it would be beneficial to take just a specific sample of women only taking estrogen, progesterone, birth control pills, contraceptives, to see really what that does in general. It's a double-edged sword because it's really important to take those pills for other health reasons besides birth control. Mm-hmm. But it is definitely linked to either high levels of estrogen dependence. There's also some research, I believe, that has suggested there's, on the other hand, endometriosis can also be linked to progesterone receptor problems. So like it's not accurately processing. I'm not sure what the right word for that would be. Like the progesterone receptors like kind of stop working almost with endometriosis, which is another interesting thing. But I agree with you that it would be important to look at that specifically, or I guess have a more intense control even with that, a lot with that, and their food choices as well. So
yeah, definitely, dude. Yeah, it seems like there's a lot to study still.
mm-hmm.
Do you think that there's I know you're in bioinformatics, or you're working with Brubaker, right?
Yeah, this is what we're doing, kind of. Yeah.
Do you know any similar research going on at Purdue, or
yeah, so,
or studying women's health in relation to their microbiota, right? As a study states, or yeah, I mean, there's so much more to do with
mm-hmm.
women's health, and looking at, okay, how does, not to be too simple, but how does certain how does one thing look in a control population with this disease? How does medication affect it? How does there's a lot of great stuff to study there. Do you know of any researchers doing that?
So like any lab specific to women's health, I don't think, like in the biomedical engineering, I from best my knowledge, I don't think they exist. I think there very well could be specific labs in some more life science related fields. I am in like kind of a subgroup in the lab I'm working with right now with women's health in general, and we're kind of we were doing some stuff with bacterial vaginosis, and now we're really kind of shifting to endometriosis. So we're going to start doing some stuff with that, which is initially got me interested in this article. Kind of gets a good overview of the problem for sure. But yeah, I agree. There is a lot of unknown. It's hard to figure out what exactly is causing all these problems. It's interesting for sure. And like not even with endometriosis, like the gut microbiota, like I said before, there's been links to so many other diseases as well. Like it's insane how much it can control things, for sure. But yeah.
Cool. But yeah, there's just a ton in this article. There's a variety of different bacteria that they analyze for.
Yeah, trying to
do you
like
think
figure out
that
to
if
present was
they,
difficult.
yeah, like they use a general sequencing technique for this
Mhm.
bacteria, but or for the microbiota in general, but do you think, I don't know, I think it'd be kind of a cool path to go where if they identify a principal bacteria that affects endometriosis, and then they can kind of see, okay, is it present in gut microbiota? Is there different levels of that? I don't know. I don't know if it's gonna be present in both locations, or I don't know how that, yeah,
Mhm.
how association would work, but I feel like if they're able to identify a specific one, right, they're not going to
Mhm.
use 16 s RNA um,
Right.
sequencing, right? They might be able to use a more specific technique to identify the levels of those
Mhm.
bacteria. But they only did the sequencing. They didn't. Do you know if they controlled for the amount or the amount of bacteria that they found in the gut, or did they just identify
Yeah, that
identify the
that was
bacteria?
the abundance, I'm pretty sure.
Okay, gotcha.
Yeah. Um, yeah, I'm honestly, so most of what I do is computational, so I wasn't super familiar with that specific sequencing technique and why it doesn't exactly account for the entire gut genome is what they talked about in their discussion.
Mhm.
But there's like, it's interesting because like there's bacteria, but then there's also, they produce cytokines, and then they give out all these other metabolites as well, which
Mhm.
I think that could be another interesting pathway to explore. If it's, is it the bacteria or is it these intermediate metabolites being produced? Really, what is it? So
Yeah, totally agree.
yeah, and it's harder, I think, to, you can't really sequence the other stuff, but you can definitely get concentrations of uh, these other, biological markers, definitely. So
Cool. That's all the questions I have. Sarah, do you have anything?
No, this is actually all new to me, so it seemed pretty interesting though. So thank you.
Yeah,
yeah, I apologize if it was a lot. It was yeah, especially the charts right there, and the stats are pretty intense, but yeah,
yeah,
it's
this is the last journal club, I believe, for the semester. So, Sarah and Nina, if you're interested in attending the journal club, you don't have to present. Or, Sarah, if you want to present at the next journal club next semester, you're you're welcome to. I think we're going to have the same frequency, so about 3 a semester,
mhm,
and yeah, it's just somewhere to
yeah,
chill, learn about new research, learn how to present research, and learn about different aspects of research. Right, this is a highly statistical uh, 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 presentation, but there's also, different imaging presentations. There's different public health presentations we've done in the past. So yeah, it's definitely a great way to learn the different parts of research.
for sure. Yeah, so it's a really good idea to have this thing started, and maybe you guys can incorporate that into club meetings a little more next year or something. So
Yeah, how to,
yeah,
but
yeah,
cool. Um, yeah, thanks Nina for presenting. It
yeah,
was
no
awesome,
problem.
awesome presentation.
Yeah, thank you, Nina.
Thank you guys for listening. Yeah. All
All right,
right. Have a
I'll
good
go ahead
weekend,
and end the
everyone.
recording. Yeah, and then I'll end the meeting as well. Thanks guys for attending.
Thank you. Bye.